and welcome to today's webcast. My name is Cole and I will be your event specialist today. All lines have been placed on mute to prevent any background noise. And please note that today's webcast is being recorded. During the presentation, we'll have a question and answer session. You can ask text questions at any time. Click the green Q&A icon in the lower left-hand corner of your screen, type your question in the open area, and click Submit. If you would like to view the presentation in a full screen view, click the full screen button in the lower right-hand corner of your screen. Press the Escape key on your keyboard to return to your original view. For optimal viewing and participation, please disable your pop-up blockers. And finally, should you need technical assistance, as a best practice, we suggest you first refresh your browser. If that does not resolve the issue, please click on the support option in the upper right-hand corner of your screen for online troubleshooting. It is now my pleasure to turn today's program over to Patrick Lowe. Mr. Lowe, the floor is yours.
Welcome to today's webinar titled Novel Applications of Multiphoton Microscopy, brought to you by Biotechniques and sponsored by Leica Microsystems. Hello, my name is Patrick Lowe and I am a senior editor with Biotechniques. In this webinar, our two speakers will describe their use of multiphoton microscopy imaging methods to dissect the function of immune cells in vivo and in real time. Following these presentations, there will be a question and answer session with the speakers, so please feel free to enter any questions into your webinar console throughout the presentations. We are pleased to have Dr. Jordan Iacobelli as our first speaker. Dr. Iacobelli is an assistant professor at National Jewish Health and in the Department of Immunology and Microbiology at the University of Colorado School of Medicine. He will describe in vivo imaging of immune cells by two-photon microscopy. Jordan?
Thank you. And so I'll give a quick introduction regarding two-photon microscopy. Two-photon microscopy is a type of fluorescent microscopy which uses infrared lasers to excite samples deep into tissue. And so the main difference compared to conventional type of microscopy is the use of different type of source for excitation. So in conventional fluorescent microscopy, you typically use lasers that roughly match the wavelength of the fluorophore that you want to excite. So for example, in the case of using GFP, you would have a laser around 488 nanometers so that the photons emitted by that laser have the right energy to excite your GFP to emit the fluorescence. On the other hand, with two-photon, you typically will use infrared lasers that have about twice the wavelength of the fluorophore that you want to excite. This is then gives you half the energy, so you need two lower energy photons that must simultaneously hit that fluorophore in order to be absorbed and permit that fluorophore to reach its excited state. And this is a very rare event, so you need special lasers that can emit very dense packets of photons to obtain a reasonable frequency of fluorophore excitation. This also makes these lasers very expensive. It's about a tenfold difference with more conventional type of lasers. And so just to give another kind of example, two-photon microscopy typically has the advantage of really eliminating out-of-focus events because you need this coincidence of these two photons to hit your fluorophore. And so since it's such a rare event, this only happens really at the point of focus, as you can see in that little dot coming from the objective on the right. On the other hand, single photon excitation has the issue of allowing a lot more out-of-focus excitation. As you can see, you get these two cones on each side of the focal plane from the lower objective. And so due to these characteristics of two-photon microscopy, there are a number of advantages and a few drawbacks. The biggest advantage is that using infrared lasers, the photons can actually achieve a much deeper penetration. In permissive tissues, you can actually go all the way up to 1 millimeter deep. And this is a huge advantage compared to more conventional type of fluorescent microscopy, where typically you were limited to about 50 or 100 microns. Also, there's much reduced scattering of your photons and also reduced background. These lower energy photons also have reduced phototoxicity. So if they're absorbed by the tissue, they only typically will give a little bit of heat, but they don't really cause phototoxicity. As I mentioned, there's also reduced out-of-plane excitation. And another advantage of multiphoton excitation is that with a single laser set at a specific wavelength, you can actually get excitation of multiple fluorophores. So you can do simultaneous imaging of various colors. The main drawbacks are that compared to more classic confocal microscopes, you have reduced resolution, both in the XY plane as well as in, to some extent, potentially in the Z plane. And there's also some limits in the combination of fluorophores that can be used at the same time. And so I'll go a little bit into the different type of dyes and fluorophores that can be used. So in a typical two-photon microscope, you most likely will have 4 different detectors, or PMTs, photomultiplier tubes. And with typical filter sets, you can actually image in a blue channel, a green-yellow channel, an orange-red channel, and a far-red channel. And so you can actually image up to 4 different colors at the same time. Typical dyes that are used are dyes such as CFSE, which is a derivative of fluorescein, or similar dyes that have different wavelengths and different colors. And these dyes will label the whole cell body and are mostly used to track the movement of cells within tissues. They're pretty easy to use. They have the limitation, however, that you have a limited time to image your cells because over time there is either turnover of the proteins to which these dyes attach to, or by cell proliferation the dye will be diluted. And so typically you have about 1 to 3 days, depending on the cell type and the dye used, to image your cells. But beyond that, the dye will likely be diluted out for successful imaging. And so that is a limitation. Also in two-photon, we often use vascular labels, where we can use inert substances such as dextran, or a protein such as BSA, bovine serum albumin, to label the blood volume so that we can see blood vessels. Also, in many cases, you can use your favorite antibodies conjugated to various fluorophores to highlight and image specific tissue components in your imaging. Another class of fluorophores that can be used is actually genetically encoded fluorescent reporters. The advantage of these reporters are that as opposed to the dyes, they don't dilute because your cell is constantly making more. And so you can follow for a long time, and there is no issue with cell proliferation. Among the most used are either ubiquitously expressed fluorescent reporters, such as in the ubiquitin GFP mouse, in which you can extract lymphocytes or leukocytes from this mouse and then separate out your cell population of interest and use those cells that are now labeled with GFP in this example. There are also some mice that have specific fluorescent proteins transcribed by a promoter that selectively will mark cells of interest. So for example, if you're interested in T cells, there's a mouse that expresses DS-red under the control of the CD2 promoter, which is relatively specific for T cells in this context. Also for antigen-presenting cells, or dendritic cells specifically, there are mice that have, for example, YFP expressed under the CD11C promoter. Another way to obtain more selective expression of fluorophores in your cells of interest is the use of Cre recombinase as a way to mark cells. And this takes advantage of transgenic mice in which fluorescent proteins such as YFP have been inserted into the ROSA26 locus, but the fluorescent protein has in front of it a stop codon cassette that is flanked by LOXP sites. So only in cells that express Cre will that stop cassette be excised and the cells become fluorescent. So by using your favorite Cre, you can then highlight specific cell populations to be imaged. And the nice advantage, as I mentioned, is that with a single excitation wavelength, you can actually excite multiple fluorophores. And so we typically use 810 nanometers, where we can actually label up to 4 cell populations with different dyes. And the example shown here, we can use VPD for the blue-violet channel, CFSE for a green channel, CMT-MAR for an orange-red, and Alexa 647 for a far-red channel. And so in this example here, from an explanted lymph node, if you could please play the video, Patrick, we can easily follow the behavior of up to 4 different cell populations. So in this example, you can clearly see that we have green, red, blue, and purple cells that can be easily tracked. And I'll go into the actual analysis and the type of quantitation that we can do for this. But before that, I want to mention a little bit about the two main modes of imaging for two-photon. And so typically, you will either image intravitally or using explanted organs. And each of these techniques will have some advantages and some cons. So for intravital microscopy, the main advantage is that you are imaging in the most physiological environment, since whatever tissue you might be imaging or organ is still within the mouse, so it's still vascularized, you still have lymph flow. However, there are quite a few disadvantages to this technique, which make it more complicated. The animal, typically in our work we use mice for study of the immune system. You will have to have a constant monitoring of the mouse's anesthesia, the oxygenation, as well as the body temperature, hydration, and overall welfare. Another potential caveat is because there is breathing, heartbeat, and depending on where you're imaging, if it's, for example, in the gut, you might have peristalsis. And all of these can induce a lot of sample movement. So it can be pretty tricky to clearly immobilize the tissue as you're imaging it. Also, some tissues can be difficult to access and can require more complicated surgery. And the surgery can cause an inflammatory infiltrate, mainly neutrophils, at that surgical site, which could change the behavior of the cells that you're trying to image. On the other hand, explanted imaging, where you euthanize the mouse and then you take out your organ of interest, for example, for immunologists, the lymph nodes are typically used. This is a lot easier to maintain because you simply take out the organ and you just have to keep it warm and bathed with the oxygenated media. There is no inflammatory infiltrate to confound things, and it's relatively easy to immobilize the tissue to minimize movement. On the downside, there is no blood circulation or lymphatic flow, so depending on the type of scientific question that you are interested in investigating, this might not be an option for you. Also, the organ, once it's explanted, has a limited time of viability. However, typically in a lot of tissues, you can keep the tissue healthy and live and image the cells in that tissue for up to 6 to 8 hours. So that's still a relatively good amount of time to study. Depending on the type of imaging that you will be doing with two-photon, some additional equipment will be necessary. So for explanted tissue, mainly you will need a flow system with a pump that can maintain the temperature of the tissue, as well as keep a constant flow of oxygenated media in the environment, which is critical for allowing normal cell behavior. In the case of intravital imaging, again, depending on the specific tissue imaged, things might vary, but typically you will need an isoflurane machine for the anesthesia of the mouse, as well as a homeostatic warmer to keep the body temperature at 37, and specific stage adapters to immobilize the tissue that you are interested in imaging. So once you have all these images, these will create very large data sets, and you'll need specialized software to allow you to analyze these data sets. Typically, we use BitPlane from Imaris. There's also Velocity from Profinerma, and some other software that can manage these big data sets. And these are complex data sets because you have the three-dimensional space coordinates of fluorescence of your cells, as well as the time element, and up to typically 4 channels of fluorescence. And so this creates a lot of data to analyze. What these software will do is they will typically search for, based on fluorescence, the center of mass of your cell. As you can see in this example, the red cells have a dot at their center, and that will be used to track the movement of the cells over time. And you'll see the software here in this case is tracing a white line as a track following the movement of your cell of interest. And then from this type of data, you can actually extract a wealth of information. Among the things that can be quantified are cell motility. So for example, the speed of the cells that are moving, and I'll go a little bit more into details of this type of analysis. You can look at the changes in cell morphology. Very importantly for immunologists, we looked at cell-cell interactions, how cells can communicate with each other, Also, their relative localization within a tissue, the cell death, and a whole slew of other parameters. More recently, fluorescent biosensors have been developed and are being used more and more to get more functional type of readouts from the cells that you're imaging. For example, the signaling of cell. Rachel Friedman and Max Krummel have developed TCR fusion with GFP that allows to look at synapse formation in vivo in T cells. Or, for example, the lab of Thorsten Mempel has developed an NFAT reporter fused to GFP, and they can look at NFAT signaling by looking at the translocation within the nucleus of this NFAT reporter, highlighting signaling. You can also look at the activation state, or for example, the effector function read out as cytokine production. And the lab of Rich Locksley has developed a number of reporters for cytokine production, such as IL-4, interferon gamma, and so on. And you might also be able to track different subpopulation of your cells of interest. So I'll now kind of switch and give some specific examples of the type of imaging that we can do and analysis that can be obtained. So first I'll start with some examples of explanted tissue imaging. So the typical setup for the experiment would be to have your cells of interest, and these might be different cell populations or control cells and experimental cells that would be differentially fluorescently labeled and then injected into a recipient mouse, typically IV. You then wait a certain amount of time, more classically about 24 hours, to allow these cells to kind of reach a steady state and distribute to the various tissues. And then at different time points, and you might treat or not the recipient animals also, you would euthanize the recipient mouse and then explant your organ of interest. And many organs have been successfully imaged, such as lymph nodes, spleen, the pancreas. For certain large organs, you can do thick tissue sections, such as the lung, the thymus, or the brain. And then as long as you keep the tissue bathed and warmed, you can analyze this cell behavior with time-lapse imaging for many hours. And so in my specific case, I've been interested in understanding the role that the actin cytoskeleton has in T-cell motility and cell-cell interactions. So I'll give you an example of how two-photon microscopy can help us look at these behaviors in an in vivo setting. And so first I'll start with some examples on the motility of T cells as well as their interactions with antigen-presenting cells for their activation. And so in this example here, this movie will show you the movement of control cells or cells that are knockout for myosin 2A, which is a cytoskeletal protein that we've shown is important for cell migration. And so again, using the software, we can track the behavior of these cells, and you might have noticed that the green cell, the control cells, had longer tracks. If you want, you can play it again, please, Patrick. While the red cells were more confined. We can take this data and then, using different types of analysis software, actually quantify and get usable data that we can do various types of statistics. And so examples of the type of parameters that can be analyzed, we can read out the instantaneous speed of the cells, their average speed, their turning angle, as well as, for example, plot the track from the point of origin, which can be a nice visual way on the top in this slide to show how they're moving and how their type of behavior is. Also, a type of readout that's often used is mean square displacement, which measures the distance that a cell goes from its point of origin over time. And so these are all typical parameters to quantify different behaviors of cell migration. We can also quantify the behavior of T cells when they encounter antigen on an antigen-presenting cell. And so in this example, we're going to have control T cells in red and, again, myosin knockout T cells in blue, as well as dendritic cells in green, which have been pulsed with a specific antigen. And so you might notice that the red control cells typically will stop and interact with those dendritic cells. If you could run it again, please. Thank you. And basically, they are talking to each other. On the other hand, some of the some of the knockout cells, even if they're in the thick of the dendritic cell network, they're kind of ignoring to some extent those dendritic cells. And so again, using Imaris software, we can now have the software generate 3D surfaces of the cells of interest. And so for each of the 3 populations, the 2 T cell populations and the green dendritic cells, and then we can extract a wealth of information. For example, we can ask what percentage of our T cells are interacting for a certain amount of time with dendritic cells. Also, we can inquire and analyze how long these interactions are, and whether there's serial interactions with multiple dendritic cells or single interactions with one dendritic cell. And this information can be very valuable for immunologists because in the case of T cells, it's been shown that typically long interactions tend to give more productive activation of a T cell, while shorter interactions tend to be more tolerogenic for a T cell. And so by analyzing this type of behavior, we can get some information on the type of immune response that might ensue from a certain kind of stimulation and how different intervention might modulate that. And so then I'll give a final example of how instead for certain type of questions you need to use intravital microscopy. And so another function that I'm interested in analyzing T cell behavior is extravasation, which is the process by which a lymphocyte is able to exit the blood flow by migrating through the endothelial blood barrier and enter a tissue. And so for this, obviously, you need intravital microscopy because you need an intact blood flow so that you can analyze this behavior. And so for this, again, the setup would be similar where you would have fluorescent label cells of interest that would be injected IV into a recipient mouse. And also we use typically a reagent such as dextran to highlight the blood vessels. Then at different time points, the recipient mouse would be anesthetized, and we would have ongoing anesthesia through an isoflurane nose cone, put the mouse on a heating pad to keep it warm, monitor its vital signs, and then with a small incision, we can open up an inguinal flap to expose the inguinal lymph node while maintaining the blood vessels to that lymph node so that we can actually image the process of extravasation. And so in this example here, you can see a zoomed out view of the complicated network of blood vessels and lymph node. So the blood vessels are in red and the T cells are in green. And then if we zoom in using time-lapse microscopy, we can then look at the behavior of these cells. And so from the zoomed in area, you'll see on the right, if you can play the video, please, you'll see this T cell in green that's in the middle that kind of moves around the blood vessel looking for a point of exit. Eventually it comes to that point, it comes out, and then as it goes deep into the tissue, we kind of lose track of it. If you don't mind, you could play it one last time. And so again, this is just a control cell, but you can imagine that you could use different types of treatments or compare control cells to experimental cells and see if the typical behavior is changed by a different type of intervention. This is just an example of extravasation in the lymph node, but many other tissues have been analyzed using intravital 2-photon microscopy. For example, the lung, the pancreas, the spleen, and the central nervous system. So depending on your scientific questions, many of these organs or tissues are accessible by 2-photon microscopy. And so in conclusion, I hope that I gave you a good overview showing how 2-photon microscopy allows for the time-lapse imaging in vivo as a way to be able to study the behavior of immune cells in various tissues in order to characterize both their overall behavior and how different interventions could alter this behavior. And I just want to finish by thanking the people that help with this work, Miriam Eston and Scott Thompson in my lab, as well as our collaborators in the Friedman lab and in the Kreml lab. And thank you all for listening in.
Thank you, Jordan, for your talk.
You're very welcome.
Our next speaker is Dr. David Fuchsman, who is an assistant professor in the departments of pathology and of microbiology and immunology at the Albert Einstein College of Medicine. Dr. Fuchsman will discuss the technical considerations for imaging plasma cells in vivo and the biological insights gained from those studies. David?
Thank you. And I also want to thank Jordan for the really thorough introduction into intravital imaging. I'll be touching on those points in a little bit more depth, specifically focusing in on plasma cells. So for today's talk, I provide a brief introduction to plasma cells. Then I'll discuss tools and reporters for imaging plasma cells in vivo and some technical considerations. Finally, I'll describe some biological insights we've obtained by imaging plasma cells in vivo. So to give some background on plasma cells, they're terminally differentiated B cells that secrete antibody constitutively. These cells are critically important for immune function and memory, as they are long-lived cells that can secrete antibody for long periods of time. I just want to point out that the half-life of antibody to viral infection can exceed 50 years, and this is independent of memory B cells, in that treatment with rituximab, an anti-lymphoma treatment which eliminates memory B cells but spares plasma cells, and in turn serum antibody also remains. Therefore, these cells are critically important for long-term immune function. Plasma cells are also important in malignancies and various cancers. Most notably is multiple myeloma. This is a malignancy transformation of a plasmablast. These tumors develop in the bone marrow, and in late stages they spread to other organs. Current treatments for these diseases, high-dose chemotherapy and autologous bone marrow transplantation, and unfortunately survival is 3 to 5 years. There are other types of plasma cell malignancies, and also plasma cells can also become a problem in autoimmune diseases as they can produce antibodies against self. So understanding the mechanisms that control their survival and their migration are critically important for both immune function and also for these pathological conditions. So back to normal plasma cells. Plasma cells can develop from a variety of sources and come in many varieties. Most classically, they arise from naive B cells that pick up protein antigen and interact with T cells, and then they can enter into a germinal center reaction where they can modify the affinity of their receptor for antigen. From the germinal center, B cells can then differentiate into memory B cells or plasmablasts, which are considered the precursors of plasma cells. These cells will migrate to plasma cell niches in the bone marrow, mucosal tissues, or in the spleen, where they can survive for long periods of time. The migration and the survival niches are poorly understood, but they're critically important for long-term immunity. In addition to this pathway, there are also plasma cells that are generated early in the response. B cells can skip the germinal center and generate short-lived plasma cells, which may be important for early stages of infection, but they may also seem to be more prone to generating autoimmune responses, more important for autoantibodies. There are also T-independent plasma cells that can arise, and these can both be short-lived and long-lived. Now, for visualizing and studying plasma cells in vivo, there are two reporters of antibody-secreting cells. Both use the master transcriptional regulator of the plasma cell lineage, BLIMP1, which is encoded by the gene PRDM1. The BLIMP1 GFP reporter is a knock-in of GFP into the endogenous locus, which can be used as a reporter as a heterozygous, or as a knockout of BLIMP1 when it's homozygous. Here on the left is a FACS plot, and expression of the GFP in plasma cells kind of peaks at about 2 logs above background. I'm also showing the CD138, which is the canonical marker for plasma cells. The second reporter, the BLIMP1 YFP, also uses a PRDM1 promoter, but it's generated as a BAC, or bacterial artificial chromosome insertion, and it uses the venous variant of YFP, which is a fast-folding variant. And as a BAC transgene, most likely multiple copies were inserted in tandem, which may explain why the fluorescence is more than a 3-log shift over background, shown on the right side. So this YFP reporter is a bit superior for imaging plasma cells in vivo for this increase in fluorescence. And the links to the papers are at the bottom. So both of these reporters faithfully report on plasma cell differentiation, but for the purpose of this talk, I will focus on the BLIMP1 YFP reporter, which I have used in my studies. So intermediate levels of BLIMP1 YFP represent a mixed population of plasma cells and pre-plasma cells containing proliferating plasmablasts. The highest expression of BLIMP1 corresponds to plasma cells, which are CD138 high. Expression level correlates with differentiation, but it's not sufficient to truly distinguish between plasmablasts from plasma cells without using additional markers. Therefore, BLIMP1 positive cells can be collectively referred to as antibody-secreting cells, a catch-all for all groups of cells. One technical consideration when using these mice is that BLIMP1 is not expressed only in this lineage, but is expressed in other lineages as well. Some endothelial cells express low levels of YFP, but they're easily distinguished by their morphology. BLIMP1 is also expressed in a subset of CD8 positive T cells. And this, in general, this is shown in the upper panels on the right. In general, the CD8 positive YFP cells are a bit dimmer than the antibody secreting cells, but there are some cells that have high expression, and so those cells can easily be mistaken for plasma cells. One workaround is to breed BLIMP1 reporter into a TCR deficient background using TCR beta delta knockout mice. In this background, CD8 cells are absent, and the YFP expression is exclusively found in ASC compartment in the hematopoietic lineage. That's shown on the bottom. The other option is to obtain high fidelity expression is to transfer T cell depleted populations of lymphocytes from the BLIMP1 YFP reporter. So we decided to image ASCs in vivo by two-photon microscopy to try to get an understanding of how these cells develop and migrate in the lymph node. To do so, we bred NP-specific heavy chain allele B18 high with the BLIMP1 YFP reporter. We purified naive B cells from these mice and transferred them into ova-immunized wild-type recipient mice. The next day, foot pads were boosted with NP conjugated to ova protein to induce YFP-positive ASCs. And we also transferred in polyclonal B cells 1 day prior to imaging. We monitored the responses from day 4 to day 10. This is a slide of an intact lymph node explanted from this type of imaging, and we're imaging here by two-photon microscopy. In green are YFP-positive cells, and you can see in the top center they've accumulated in a region called the medullary cords. In orange dotted lines, I've marked B-cell follicles that are kind of on the periphery of the lymph node. And in these blue ball masses, those are germinal centers. And the red marks the follicular dendritic cells that are present in the light zone of the germinal center. So we image the migration of these ASCs from the germinal centers and the B-cell follicle where they accumulate in the medullary cords. So Patrick, can you play this video? So here in green are BLIMP YFP cells, and what we found is that these cells migrate in a very linear manner in the lymph node, but in different directions, kind of randomly oriented, in contrast to the naive B cells in blue, which are kind of undergoing a random walk characteristic of lymphocytes. We can even see them migrating towards germinal centers and away from the medullary cords. And here, right when they're very close to the medullary cords, we can still see cells migrating away from the structure, suggesting that they're not migrating in some type of chemotaxis, but more of a random migration, even though they accumulate in this structure. This migration is also very unique to plasmablasts or antibody-secreting cells. We don't see this in normal lymphocytes or other lymphocytes that have been characterized in the lymph node. While I should also point out that in the medullary cords, these YFP cells are arrested, although they are jiggling a little, which I'll talk about later in the second part. As Jordan described, we can analyze their migration and extract information. We track their track velocity, and what we see is that the BLIMP YFP cells are migrating a bit slower than naive B cells in the lymph node. Although their migration also decreases as they reach the medulla, the medullary cord. We also tracked their mean square displacement over time and found that in contrast to naive B cells, which undergo this random walk and their displacement is linear over time until they peak as the size of the follicle is limited. The Blum-1-YFP cells undergo this precessive walk, which shows this upward curving migration that's seen in the green line. And the ones in the medullary cords are arrested, and they don't displace at all. We also analyzed the displacement directionality by analyzing the movement in the X, Y, and Z planes of a variety of movies. And what you can see that both naive and Blum-1 positive cells have the average of their displacement in any direction is close to the 0 mark on the axis, indicating that there's no directionality. So they move in a precessive manner, but they don't seem to have a direction in terms of where they're moving. Can you play this video twice? So because chemokine signaling is so important in the movement of lymphocytes, we asked whether pertussis toxin, which blocks chemokine signaling, most chemokine signals, has an effect on Blum-YFP positive cell migration. And what we found is that naive B cells in blue arrest completely, while the Blum-YFP cells continue to migrate, although a bit slower, the green cells. So you can see that the green cells continue to migrate even though the blue cells are pretty much all arrested. So we can quantify that again using the tracking software, and what we see is that naive B cells in black are completely arrested after PTX or pertussis toxin treatment, whereas the Blum-YFP positive cells are slower, but they continue to migrate. And that can also be analyzed by mean squared displacement, where naive cells are completely arrested in red after PTX treatment. The Blum-YFP cells, although are slower and displace, takes more time for them to displace, they continue to migrate in the B and T cell zones, but not in the medullary cords where they're arrested in either condition. We also asked whether if they're responsive to chemokine signaling in vitro, we used the chemotaxis assay, which I won't go into the details, but basically Blum-1 positive cells do respond to the chemokine signal CXCL12 or SDF1, but if they're pretreated with pertussis toxin, they're no longer responsive, which indicates that although these cells can use chemokine signaling, they do not require them in the lymph node, which is a surprising finding and something you wouldn't have expected based on previous studies. So this is summarized here. Also, from our results we described that ASCs undergo a random precessive walk in the B and T cell zones in the lymph node, eventually finding the medullary cords where chemokine signal is critical for the retention. If you'd like to read more, you can go to this link. Once these cells reach the medullary cords, they interact with myeloid cells and stromal cells. These interactions are thought to be critical for ASC differentiation and survival. We were interested in visualizing these cell-cell interactions using fluorescent reporters for various cell lineages. One such mouse that was particularly useful was the LysM or lysozyme M GFP mouse. In these mice, DCs, macrophages, and neutrophils are fluorescently labeled in GFP. If you play the movie now twice, what we can see from these movies, here the Blum-YFP cells are in red actually. You can see that the cells are interacting directly with the GFP network that's in orange. And in fact, in the middle, you can see two red cells pulling on a GFP cell, which it's often tricky to know if cells are actually interacting by movies because you can't see the dark space. You don't know what other cells are there. But when you see cells pulling on each other and moving in response, it's quite unequivocal that the interaction is specific. What's also quite striking is that wherever there's a Blum-YFP cell arrested, they're in contact directly with a network of GFP cells, whereas naive B cells in green are migrating through this network and not really interacting with either cell type, which highlights one of the most important aspects of time-lapse imaging and intravital imaging is that without time, you can't really resolve which interactions are dynamic and which ones are actually stable and critically important for immune function. So I'll just point out that we went on to study the function of these myeloid cells in terms of ASC function. We were surprised to find that removing these myeloid cells actually increased ASC responses. So these cells are actually inhibitory in their function. If you'd like to read more, you can go to this link at the bottom. Underlying the myeloid cells in the medullary cords is a network of stromal cells. The chemokine receptor CXCR4 is highly expressed on ASCs and is critical for the retention in the bone marrow and positioning in the medullary cords. Using an SDF1 or CXCL12 reporter, the chemokine for CXCR4, shown in red, we can see that the medullary cords are highly enriched for the SDF1 producing cells. ASCs are directly in contact with these cells and migrate along them. So in addition to visualizing migration and cell-cell interactions, we can also use intravital imaging to study cell survival in vivo. ASCs are highly susceptible to apoptosis and require cytokines such as IL-6 to survive. We found that the canonical marker of ASCs, CD138 or Syndican-1, is required for ASC survival in an IL-6 dependent manner. Measuring apoptosis in vivo is quite challenging since apoptotic cells are rapidly cleared by phagocytes. However, by intravital imaging, we were able to capture bona fide apoptotic events in real time by visualizing blooding and then quantifying them. We found that Syndican-1 deficient ASCs had higher rates of apoptosis in vivo as compared to wild-type ASCs, which confirmed our ex vivo results as well. The details of the role of CD138 can be found in this reference below. Finally, arguably the most interesting tissue to study ASCs is in the bone marrow, where long-lived ASCs are found. Most labs that image the bone marrow use the flat bone marrow found in the calvaria or skull. We developed a surgical preparation to expose the medial region of the tibia. To enhance resolution, we thin the bone using a drill and stabilize with a metal plate. Using this prep, we can detect up to 150 microns in the bone marrow and detect ASCs intact bone for up to 8 hours. And here's some pictures of ASCs, and the bone is shown in purple at the top. This is a schematic for the preparation. Here we've generated a bone marrow chimera using PCR-deficient BLIMP-YFP bone marrow to visualize ASCs in an SDF1 reporter. You can see that SDF1 cells are highly concentrated in the bone marrow, not rare population as some have reported. ASCs are widely dispersed throughout the bone marrow. But in contrast, malignant myeloma cells generate these cluster growth or foci, which you can see on the bottom. These still images were taken from live anesthetized mice, and we are working better to understand how the bone marrow is supporting the survival of these normal and malignant ASCs, as we have done in the lymph nodes. Just to conclude, I'd like to thank the members of my lab, our collaborators, and our sources of support. Thank you.
Thank you, David, for your presentation. We have now reached the end of our presentations. Thank you to both of our speakers and to Lakeland Microsystems for their sponsorship of this webinar. We now have some time for our presenters to answer and discuss selected questions from the audience.
To ask a question via the web, click the Q&A button in the lower left-hand corner of your screen, type your question in the open area, and click Ask to Submit.
Hi. We have several questions. One question is, I think both Jordan and David could try to answer. Can two-photon imaging be used on already frozen tissue after sectioning and prepared slides, especially brain sections? Would that be a problem? Jordan?
So the main issue is that once you freeze or fix, then you're going to lose the time element. Your cells are not going to be alive anymore. So you can definitely image and get the depth from the two-photon, but you're going to lose the kind of dynamics aspect. So if you're just interested in depth, probably just doing serial sections, going deeper, might be an easier solution. Otherwise, people have imaged neuronal tissues that have been explanted in thick sections and have kept alive without freezing, so that you can actually still look at cellular behaviors and dynamics.
Great.
The other thing I'd point out is one of the nice features of two-photon imaging is you get the second harmonic generation, which can give you information about collagen or repeated structures. And even with sections, you can get that same information, and that can be very useful, even if you're not doing in vivo imaging.
Great, thank you. Another question then is, can two-photon microscopy be used to monitor calcium flux in neuronal cells, for example, after the cells have been exposed to a certain treatment?
Yes, so that's a very active area that I don't think either of us touched on, but there are a variety of both calcium dyes that can be used or genetically encoded reporters like these. There's a whole family of GCAMP proteins which can report fluorescence in response to calcium, and there are mice that express these that can be used both for studying questions of neuroscience or in immunology or in other fields as well.
Great. Then another general question is then, can two-photon microscopy be currently used for drug screening related to toxicology? Do either of you know this?
Yes, I think that's been used for certain instances. If you're looking at, for example, cell death or inhibiting tumor migration, there are examples of use of two-photon microscopy for that. Obviously, you probably need to do it more longitudinally over, potentially days. So you need a very specific type of imaging technique. There are certain labs that apply what people call like an imaging window. So you fix almost like a glass cover slip to a certain area that allows you to go back over days, so that the mouse can then be woken up from anesthesia after an initial imaging session, and then a day later or a couple of days later you can go back and try to image the same spot. For immunology, we don't use that as much because the immune cells are so dynamic, so that the specific cells will move far away out of the imaging part that you're looking at. But for tumor cell migration, so tumor metastasis, that has been done. For example, the Condilis lab has done a lot of that.
Great. And then another question is, what is the penetration depth of the multiphoton microscopy? Can it be used to image cells in all locations of superficial lymph nodes, superfacial lymph nodes?
So typically people say that the maximal penetration is close to a millimeter, but of course it's very difficult to always get that kind of penetration. And a lot has to do with the image quality, the surgical quality, the amount of light scattering in the prep based on just the type of tissue. Lymph nodes tend to be pretty good for having minimal light scattering, but they're just from a bad prep can also reduce your visibility or issues in your light path. But usually the technical limitations of the microscope are never reached, but more the practical aspects of trying to visualize because of the prep or whatever are usually the limiting factors.
Great.
Yeah, I would say, as you just said, probably a millimeter is your best kind of scenario, and that's more for neuronal type of tissues, which don't scatter as much. But for immunology, like lymph nodes and particularly like the spleen, scatter much more. So you're probably more limited to, between 300 and 500 microns would be my guess.
Okay. Our next question then is, what laser wavelengths are you using when you excite for different fluorophores?
You want to go first?
I can. Okay. This is Jordan. For our work, so when we use dyes, so the chemical dyes, typically we prefer 810 nanometers because we can get good excitation of all those 4 dyes that I mentioned. For example, the violet proliferation dye, CFSE. We can also use something like eFluor 670 and CMTMR. When we're using genetically encoded proteins such as GFP and red versions of that, typically we go at a higher wavelength, so more in the range of 910 or so.
Great. David, do you have anything to add to that?
I would agree. I mean, there's if you need to access some of the far red like M-cherry, often you need to like a
an OPO to reach higher wavelengths, like in the above 1,000, like around 1,200 or something. But you can also sometimes reach them at very low wavelengths, like around 760. We typically work around between 880 and 920 because we use mostly genetically encoded fluorophores, and usually you can capture most of them with the single wavelength, as Jordan was explaining earlier.
Great, thank you both. Well, that is all the time we have for questions. I want to thank all of you who have joined us today for this Biotechniques webinar on using multiphoton microscopy for studying immune cells in vivo, and we hope that the webinar has proven to be informative and useful. I'd like to once again thank our speakers for their presentations and Leica Microsystems for their sponsorship of this webinar. Goodbye.